idea why it's doing that. What is that thing? I don't know. Go away. It won't go away. Please move this window away from the shared application.
Oh.
But then it comes back. I swear, but all my USB ports have crapped out on me. I can't use my USB ports. I go home now, I can't project it to my home screen at home. It's possessed. My laptop is just possessed. Yeah, I do. And then I call Micro Repair and they're like, well, it's a 4-hour turnaround time if you want it fixed. Who has 4 hours without a computer at this time? Me. So, all right. Seedless vascular plants. What do we want to know? I don't know why it's doing that, but oh, that's not what I want.
What are all the phylums we need to know for this one?
So we have okay, so let's go to the last slide where it has all that table. So asterisked ones, so the ones here, Lycopodiophyta, Pteridophyta, whoops, Polypodiophyta, sorry. The phylum Lycophyta, Polypodiophyta, Psilotophyta, and Equisetophyta. So the ones that I've typed in, I think are what your book uses as well. Lycophyta, Polypodio, I folded that one. Psilotophyta, and then I'll use Equisetophyta.
Did you want us to know like the miscellaneous characteristics of them as well?
No. No, it's mainly looking at, do they have root stems and leaves or not? Types of leaves, and then type of steel would be useful. So 3 of those columns, root stems and leaves. Do they have those? And if they do, what kind of leaves? And then steel type. And then, so with the seedless vascular plants life cycle, it's just the idea of, you know, the generalized alternation of generation. So remember, the sporophyte is the dominant now, moving forward from the seedless vascular all the way through to angiosperms. But I also showed you life cycles, you know, like the whisk fern, but it's basically the alternation of generations, right? I'm not going to pop up something on here and ask you to tell me about it. However, knowing what a whisk fern looks like, yeah, that'd be a good idea. Whoops, I didn't mean to do that. So the basic identification of, you know, what does a club moss look like? What does a horsetail look like? And their phylum names, which organisms belong into which phylum. Certainly, I'll be asking that kind of stuff. Economic importance or why we would use them. Those are always good, especially for essay questions. Do you have specific questions on the seedless vascular plants? Certainly know what strobili mean.
So for the ferns,
Yeah.
is it strobili or spora?
So good question. With the ferns, they don't have strobili. They have sori. They got to be different. So there we look on the underside of their leaves for the sori. So that's what you're seeing here in slide 57. That's under the microscope, so that's the leaf of the fern, and then this whole cluster, this entire thing is a sorus. And it's just a cluster of sporangium, so all of those are sporangium tissue all clustered together. But it's called a sorus. Other questions on the seedless vascular plants?
Like
Yes.
a.
Yes. So know the organisms, and so with the Lycophyta, club mosses, spike mosses, quillwort. I'll probably refer to them that way rather than their genus names. I'll use their common name, that's a little easier. Any other questions on this set? Well, then we did gymnosperms, right? After that. So as you are studying this, it's a good idea to look at these essentially three groups, the seedless vascular, the gymnos, the angios, and kind of think of like, what's similar, what's different? And so obviously jumping from the seedless vascular into the seed plants, it's a huge difference right there. The fact that the gymnos now are producing seeds as their unit of dispersal, they don't release spores anymore. So looking at, the slide 7 here, where we're getting into what is so great about the seed. And then slide 9 is really the only time I'm gonna get you to think about bryophytes for the final. Thinking about how the bryophytes and the seedless vascular plants, they release spores as their unit of dispersal. But gymnos and angios release the seed. So this slide, I think, is pretty important to think about, and then use this as a comparison to, okay, what's going on with the gymnosperms and the angiosperms. Know the phyla, the 4 phyla, and who belongs into each one. Then we got into the Pinus life cycle and how the female part, the ovule, which is really thinking about the megagametophyte. But that megaspore no longer gets released, it gets retained. So the megasporangium never opens up, never releases the spore. It just keeps the spore in there. You just have one, the megaspore mother cell. And it still goes through meiosis, you still have that, but then only one of those remaining cells, or those 4 cells from meiosis, make it. So that's why they call it the functional megaspore. So it's held within the megasporangium as it goes through all of that, and then the functional megaspore has to go through a bunch of mitosis and growth to develop into the megagametophyte. So from shifting from slide 14 to 16 here, you see the development where that megaspore went through lots of mitosis and development to get you that megagametophyte. Still within the megasporangium and even further protected by the integument tissue. So when you look at that, this is like a mature ovule ready to go, ready to get pollinated. On slide 16 there. So I'll definitely be asking about that, looking at that progression.
As in like the Pinus life cycle or just mainly the?
Well, the ovule, yeah, I'll be asking for ovule details on how does this work. And then the Pinus, I mean, this is part of the Pinus life cycle, but I want you to see the difference here, how everything's retained and withheld. And you get the megagametophyte held in there on the sporophyte tree. And then, here it's just waiting. It's got the eggs ready to go. And now it just needs to get pollinated. And remember, pollination, it's a different definition when you think about it in gymnosperms. What is the pollination definition? Transfer of pollen to what? Not the stigma. To the ovule. Yep, to the ovule. Transfer of pollen to the ovule. So that pollination droplet's right there. It's like, okay, I'm ready for the pollen. All right, so it's this mature ovule. It's ready to go. And now, this is really in the cone. If you think about a typical pine tree, for example, a Pinus genus, this is all happening in the cone tissue, female cone. And we also got into the male, what's going on in the male cones or the pollen cones. That they have 4 cells, but when they get shed out of the cone, they're still immature. There's no sperm developed yet. As far as the timing, we went through this. I'm not gonna ask for details, like tell me what happens at 1 month, 13 month, 15 months. Okay, I'm not gonna do that, but knowing the. I wanted to touch that screen like that was gonna do something for me. All right, so, the time between pollination and seed dispersal is about 18 months. That number I'd like you to know. But I thought it's just really interesting to see this very slow progression of how everything goes but I'm not going to ask you to memorize step by step on that. But this is a key idea. 2 sperm are discharged, but 1 dies. Only 1 makes it. So again, that's a difference compared to what we see with the angiosperms. So compare and contrast as you're studying, thinking about how things are similar, how things are different. Why is it different? What advantages might it give them? What other questions? Yeah. Needle stuff. Yep. Is that on this slide set or the next one? I think it's the next one. I'll keep this one open, but I'll open up the other one too. So technically, the pine needles aren't true leaf tissue. They're modified branches. I'm okay. You tend to just say pine needle, keep it at that, but knowing that initially when they're babies, they just have single needles, and then eventually after a couple years, typically, they start to group their needles with new growth. And then that fascicle, so then that fascicle term comes into play. And technically, that's the paper thin true leaf, is that little brown thing, but anyways, that's how they're clustering once they're a couple years old. And then we went through, the Pinophyta is definitely the largest group. And then, but we also looked at some of the others. So, knowing who belongs into which phylum. And just like being able to recognize a, a typical specimen for those would be good. So everything, in this, there's pictures here that are good for thinking about, what do they look like? What is that? And then ginkgo, which does not look at all like a gymnosperm. And then that funky phylum Neophyta. So I guess this one I will be asking by genus name. So I'll use Needham ephedra welwitschia. If I talk about those organisms. The table at the end. So knowing if they have tracheids or tracheids and vessels, that's kind of weird. So remember, tracheids are the more primitive type of xylem. And gymnosperms have that, except for the needlephytes. Groups in there, the needle. Types of leaf produced would be good. Okay, what other questions do we have on the gymnosperms? Yeah, the pine needles that there's a little bit recessed or sunken. Yeah. That's a good adaptation because their needles persist for so long and then although around here, they often go dormant in the winter, but sometimes on a nice winter day, they'll reactivate and they'll keep doing their transpiration. Any other questions on gymnos? Yeah.
The gymnosperms, or this could go for angiosperms as well, but are the gymnosperms the one where the two like sperms both do something and they like fuse and one does like the outer and one does like the inner?
So with gymnos, that's a difference because we have two sperm that will eventually get produced. But with the gymnos, one will fertilize the egg and the other one dies. But with the angiosperm, one fertilizes the egg. What does the second sperm do?
The end.
Yep, you got it. It fuses with the polar nuclei and that leads into the development of the endosperm. So that's a huge difference. So again, this idea of comparing and contrasting between, what's similar, what's different. And also the gymnosperms have in each pollen grain, there's 4 cells. Do you remember how many cells are in the angiosperm pollen grain? 2, just 2. So the angiosperms simplified. They're like, let's just go down to 2. Well, let's look at the angiosperms. So for sure, knowing the anatomy of the flower, except for peduncle.
We should have to know that. I think that's a bonus question. It's
Yeah,
ingrained in my.
I
mind
know.
since you said
It's
it
just-
that One day.
I rarely said it out loud ever. And so the first time I said it out loud, it was pretty much this semester. I'm like, oh, it's kind of a fun word. Yeah. So flower parts for sure, and then embryo sac parts or anatomy, so definitely be asking about that. So that's slide 15. So really that's the megagametophyte, but they tend to call it an embryo sac. And I'm only asking for the polygonum type, which is what's shown on slide 15. Interesting. So obviously like slide 2 where it's getting into the, the gymnosperms are naked seeds, the angiosperms are encased in fruit tissue of some sort. Ovary position, what's the significance of that? We tend to see that shift as it's being shown there in slide 7, superior, half inferior, inferior, as the plants become more advanced. So, and then we looked at, again, you have that megaspore mother cell going through meiosis, but only one survives. So then you have that functional megaspore retained within the megasporangium, and again surrounded by the integument. And then instead of doing all that crazy cell division like the gymnos do, they just said, let's just do 3 rounds and call it good. And then they migrate their cells to different parts of the embryo sac. And then you end up with 8 nuclei, 7 cells. So it's a little grainy, I guess, but we're trying to show how there's a central cell that have those 2 nuclei. Whoops, oh, it won't let me. Ugh. I can't draw when it's big, when I zoom up or zoom out, zoom in. And this is really annoying. So if you look at this, I don't know if you can see, they're trying to make it kind of green right here. So that's your central cell with the 2 polar nuclei. And then it makes sense that the egg is closest to the opening, so the sperm has an easier time getting there to the egg. But both sperm will participate in doing important stuff. One has to fertilize the egg and the other one fuses with the polar nuclei. So with the polygonum type, you have pretty simple. But, be aware there's some other options. Hmm. What questions do you have on angiosperm? Five, do you want us to know the varying ovule position or? No. Okay. I just put that in there to show you that there's options. Yeah. And we actually use the word placenta. That is interesting.
Does it serve as the same,
Mhm.
like in terms of
Feeding.
nutrients for the,
Yeah.
yeah,
I was surprised they kept that term.
yeah, it's very new
Mhm.
legend.
Yeah, usually they like to separate and make unique terms and make us memorize more stuff, you know?
Right?
Things, because they don't want you to get it obviously with animals, but then there's things like where it's like a cardboard and it's inside of it, this one is.
Yeah, exactly, right? There's like no rhyme or reason.
And then it's like we're avoiding eyes and because you don't want to get into that fight.
No, yeah, that's exactly right. And then, so much of it's Latin and Greek, and that we don't talk Latin and Greek anymore, so.
That's maybe. Like you guys talk to people who don't speak
Yes,
English.
it's a common
Uh,
language. Yep. Everyone understands the species names, what they mean. Pollen grains, now getting into, differences between a monocot and dicot, for sure I'll be asking those types of questions. Like, we learned with monocot, you have one aperture on the pollen grain. With dicots, it's 3, same with flower parts. You remember what dicots have? Multiples of what for flower parts? 4 or 5. What about mono? 3. So that must be in the second slide set. Yeah, so we got into like slide 11 and 12, a nice comparison. So make sure I'll definitely be asking about that. And then with this, knowing that the basal angiosperms are the most primitive. Basal is referring to where it's located on the phylogenetic tree, so it's a more primitive. If you go back to the slide before it. It's lower down on the tree, so to speak, closer to the Luca last universal common ancestor. And then, let's see, what else on that? So this, looks at the pollen opening, seeds, what's going on there at the cotyledons inside the seed, what's going on with vein pattern. So this is a lot like those kind of nicer visual things we saw, more like that, pretty much getting into the same idea. We looked at ancient or more primitive plants, more advanced plants. I'll definitely be asking about characteristics on that. We got into a little bit about where they think the flower developed from, how that came about. Definitely need to know inflorescence. What does that mean? What other questions do we have on the angiosperms? Either slide set on this one. Part 1 and part 2.
I have a question on mono and dioecious.
Yeah,
Monoecious is just either a female or a male, but on one plant.
one plant. You got it.
Dioecious is like a both.
No, dioecious means 2 houses. That means you have a male house, you have a plant that just has male flowers, and then you have a female house. And it's a separate plant that only has female flowers. So it's 2 houses.
The monoecious would both have like a stigma and like the anthers.
Nope. So monoecious, so this is a little confusing. Monoecious means 1 house for imperfect flowers. Think about corn. Corn. Corn is monoecious. So it has a male flower, the tassel, and then the female flower is the cob, so that's 1 house, monoecious. Corn is monoecious.
Then a perfect flower.
Yeah. And then we don't use like monoecious or dioecious for that because they're perfect. So
Right.
it doesn't work. Yeah.
Yesterday in lab, I made a fruitless strawberry.
You did?
Sat there.
Yeah.
Yes, you took out every single one.
And then did you go, hey, Duncan? It's
Hold on, looking at it.
It's naked, it's like all without color.
Yeah. Yes. So monoecious, dioecious, in the angiosperm world, if you say, if you're talking about angiosperms and you're like, oh, that's a monoecious or that's a dioecious, it only works if you're thinking about imperfect flowers, which means they're either a male flower or it's a female flower. If it's a perfect flower, that doesn't work. Okay, so so let's get into the flower and fruit slide set as well. That's, I think, what we're zooming in on. Yeah, so slide 10, imperfect versus perfect. And then monoecious, dioecious. Yes.
We care about regular versus irregular versus, oh, yeah, I do have to care about regular versus irregular.
No. What was that made me think of, what was I trying to?
Do we know like pollinator types, all of that?
Yes, so I'll be asking questions like, this plant has these or flower maybe has these properties. What do you think? Who would pollinate it?
Okay. Like bumblebees. and
Yeah.
stuff, or-
Yeah, so make sure you're learning what bumblebees like, what bird, you know, like the hummingbird, maybe bats, you know, nocturnal versus day. Yeah, strong peduncles, yeah. Mm-hmm.
Be a good t-shirt.
They're botanicals flowers, and that's strong.
On the bottom of like the part one of the angiosperm set, do you want us to know like majority of like the seed formation, like in terms of their structures?
Seed and net, so I want you to appreciate slide 54 is showing that sometimes seeds do have an endosperm, sometimes they don't. Sometimes they have both an endosperm and cotyledons. But there's some variation there, you know, often when you're first taught about seeds, they always like seeds have endosperm, and it's like, well, yeah, sometimes. But in a botany class at the college level, appreciate that sometimes you'll have the cotyledons kind of take over that role and the endosperm disappears as the cotyledon matures. And then sometimes you have both endosperm and cotyledons. But I won't get into any of the like the terminal like hypoconal, epiconal, no. That is more just to get you to realize that the cotyledons are there somewhere. It's just sometimes they're under the ground and in the soil. Um.
Rough estimate on how many questions.
Right now, it's sitting at 44 multiple choice.
Thank you.
And yeah. What you're expecting, like 70?
70, yeah, the usual.
Yeah, well. Because right now, I don't think it's gonna get too much bigger than that. And then essay, I think right now there's like 4 or 5 questions. So it's kind of pretty similar to the last exam, I think. Then we got into fruits. Make sure you know the like slide 4 and 5 and 6 in the second, the flowers and fruits 2. In that slide set, 4, 5, and 6, those definitions I'll definitely be asking. And then a lot of this it was like some of these remaining slides that show all the different fruits. Remember, I'm not getting into like any of the dichotomous key details. I was just putting these up here to show you. And then dry fruits, knowing what that means, like the, slide 2 here where it's showing a fleshy fruit. And then it's saying, if you have an exomesoendo that, really gets reduced and they all fuse together, then we start calling that a pericarp, which is a dry fruit idea.
Do we have to know that like the definition of like a fruit is going to be like beginning flowers?
Yes.
Or pollinating flowers?
Yes, definition of a fruit. And so it's in here somewhere. Didn't I put it in here? Oh, this is slide 2. So slide 1, flowers and fruits, slide 1. The definition's in here somewhere. We got into pollination. I guess it's not in this one. Where did I put the definition? It's in here somewhere. Oh, there, slide 5. Slide 5, the very title, fruit equals a mature ovary, plus or minus additional flower parts. Yes.
Example, what's that frog called that pollinate flowers? Told my roommate about it. It's like, I can't remember what the name of it is. It's on the other slide.
Do you remember, Forrest, what it?
It's something. No, I don't know. It doesn't have a comment.
In the
That's,
Xenohyla truncata tree frog. It doesn't really have like its own.
No,
Hmm.
because they're from a place where there's a million frogs, so they don't look like me.
If you go into this article, they might have a little bit more info on it, but. It's so cute. Yeah.
Here.
Well, the thing is, I really like the fruits from this plant called the milk. Really be a milk, but the problem is there's already milk tree frogs.
Oh. So
The dairy frog.
that name's taken already. Yeah.
Because those are also, there's something called an Amazon milk frog, and it's very cute. It's like, oh, this is that would be a good name for this. But already taken.
Already taken. Okay, so we have until next Thursday, noon in this room, okay? I will be available for meetings. If you want to ask questions, you can send me email, okay? So I'll be around, and so send me an email or whatever if you want to set up an appointment. But hang in there. Hang in there.
Oh, wow.